Let us consider pentose phosphate pathway, which also has another name. It is also called hexose monophosphate shunt because actually what it does, it utilizes glucose 6-phosphate that is an intermediate of glycolysis. So it kind of shunts glucose 6-phosphate away from glycolysis. As usual, we will use our outline to consider cell localization purpose, the amount of ATP or NAD plus and NADP each, number of reactions, regulatory points, and details of each reactions. So for pentose phosphate pathway, it operates in the cytosol of cells of different tissues, for example, liver, adipose tissue. The purpose of this pathway is to produce NADPH and ribose 5-phosphate in order to supply those reagents for anabolic reactions. And ribose 5-phosphate, as we will see, is needed specifically for nucleotide biosynthesis. As side products, we can consider intermediates of glycolysis, which will be also the products of pentose phosphate pathway under certain mode of the pathway. So next, let us consider the number of ATP used or released. And actually here we don't need to spend ATP for this pathway, and ATP is not released as a product of this pathway. So no ATP is involved in pentose phosphate pathway. As for the reduced equivalents, such as NADPH, here we produce 2 NADPH per 1 glucose 6-phosphate molecule. There are 8 steps total, and the regulatory point for this pathway is the first committed step of the pathway, such as the one that is catalyzed by glucose 6-phosphate dehydrogenase. Now we are going to go over the details of each reaction, but before we proceed, let us take a look on the net equation for this pathway. Here you see the balanced equation, and in order to balance it, we need to actually start with 3 molecules of glucose 6-phosphate. So since 3 molecules of glucose 6-phosphate contains total 18 carbon atoms, in the products we need to release 3 carbon atoms in the form of 3 CO2 molecules, and the remaining 15 carbon atoms will be distributed between 1 glyceraldehyde 3-phosphate molecule and 2 molecules of fructose 6-phosphate. So those 2 are products, carbohydrate products of pentose phosphate pathway, and of course we know they can be used for glycolysis because they are also intermediates of glycolysis. So for those 3 glucose 6-phosphate molecules, we need to consume 6 molecules of oxidized nicotinamide dinucleotide phosphate, and as a result we produce 6 reduced NADPH molecules. So let's take a look on the structure of the products of this pathway. We'll start with NADPH. So the structure is shown here. Again, it's considered to be a dinucleotide because here we have phosphorylated adenosine phosphate, and we have nicotinamide moiety right there, and we have those 2 nucleotides, so to say, connected to each other via a pyrophosphate bond. So the structure is pretty similar to the structure of NAD plus or NADH that we discussed before. The only difference is that NAD plus needs to be phosphorylated to become NADP plus, right? So NADPH is considered to be the currency of reducing power, and it is used mainly for anabolic reactions. And for example, we will consider biosynthesis of fatty acids or synthesis of nucleotides. And it is also used to maintain reducing atmosphere inside the cell. Specifically in red blood cell, glutathione, this tripeptide, which you can see here, needs to be always in its reduced form with the reduced thiol group of cysteine. And in order to keep it reduced in the atmosphere where you have oxygen, right, you need to have a lot of NADPH that helps keep glutathione reduced. So NADPH that you can see here and NADH, which is a product of glycolysis, are not interchangeable in the cell. They perform different functions. NADH is used as a donor of electrons for oxidative phosphorylation. NADPH donates its electrons for reductive biosynthesis. And so it is reflected in the ratio between the oxidized and reduced form of each electron carrier in the cell. In the cell, we have higher amount of NAD plus in comparison with NADH, but as for the phosphorylated counterpart of that, it's mostly present in the reduced form because it functions in its reduced form most of the time. So another product is ribose 5-phosphate. You can see it here. It is needed for biosynthesis of nucleotides and some nucleotide cofactors, as you can see here. And of course, in rapidly divided cells, such as in bone marrow, skin, intestinal mucosa, or tumor cells, anabolic processes, and specifically synthesis of nucleotides for DNA and RNA synthesis, are very active. And so you need to have a lot of this precursor of nucleotide biosynthesis. That's why for such cells, pentose phosphate pathway is very active. The flux through pentose phosphate pathway is high. So intermediates, some other products that we mentioned, fructose 6-phosphate and glyceraldehyde 3-phosphate, they are intermediates of glycolysis. And they are produced from ribose 5-phosphate or its isomers in order to preserve carbon atoms if ribose 5-phosphate is not consumed for anabolic purposes, right? In order to regenerate those carbons and use them for energy purposes instead. So let's take a look on the outline of the pathway. So here we have those 8 steps, right? 4, 5, 6, 7, and 8. But as usual, we want to split the whole pathway into some stages that can be united by some common purpose. Similarly to that, for pentose phosphate pathway, we can split it into oxidative stage, and by its name we can tell that here is where all these oxidation reactions will happen, right? So all NADPH which is produced in the course of pentose phosphate pathway is produced during this oxidative stage of the whole pathway, which includes the first 3 steps of the pathway. And the remaining 5 steps will not involve oxidation reduction reactions. They are called, they belong to non-oxidative stage of the process. And we can further subdivide this non-oxidative stage of the pathway into 2 sub-stages, so to call. So we can distinguish those 2 steps, step number 4 and step number 5, because here is where we have isomerization reaction happening. So stage 2, we can view those 2 steps, isomerization steps, as stage 2 of the pathway. And the remaining 3 steps include only the reactions involving cleavage of the CC bond and reformation of the bond to rearrange those carbon atoms between different carbohydrates.